Okay, good afternoon. And today, so we have a guest, Professor Rudolf Podgornik, and welcome to Wenzhou Bio Soft Matter channel. And I'm really happy that you came as a guest. You're actually the first guest of this channel. Thank you very much. And today I would like to ask you several questions.
Sure.
Some of them are scientific, some of them are a little bit private.
Sure.
But I hope you can explain your career, which I'm very interested in.
Sure.
Okay. So first of all, so could you briefly explain your academic career? Yeah. Yes.
Yeah. I studied physics at that time. It was referred to as the technical physics, and I was an engineer of physical sciences in Slovenia, in Ljubljana.
Mm-hmm.
Yeah. So I got my bachelor's degree there in technical physics, and then I proceeded to do my master's degree in biophysics.
Biophysics.
At the Institute of Biophysics of the Medical Faculty.
Medical faculty.
Ljubljana, yeah, the School of Medicine. And after that, after becoming a master of biophysics, I continued with my PhD studies, postgrad studies, again at the Department of Physics, University of Ljubljana. And this was theoretical physics program. So I finished in, I think it's a long time ago, 86 I think, or maybe 85.
So who was your supervisor?
My supervisor was for the master's degree. I had two supervisors. One was Professor Sasha Svetina. Yeah, and Professor Boštjan Žekš. Both of them you probably know the names from membrane biophysics and elasticity and shape equations and so on. But I never worked on that particular problem. I did other things. And as for my PhD, my mentor was Professor Žekš. And the topic of my PhD thesis was the structural interactions in the interactions between macromolecules. Yeah. So that would refer to like the structure of water, non-local dielectric response, and then I was trying to figure out what the interactions are between different molecular surfaces, like electrostatic interactions, van der Waals interactions, and other interactions. So this was my primary field, macromolecular interactions, and this was also my introduction to the DLBO theory, to the Poisson-Boltzmann theory, to the Lifshitz theory of van der Waals interactions. That all was a topic of my thesis.
I see. Okay. And you finished your PhD at which time?
So I finished, in I think 85 in 86. So I was looking for a postdoc and wrote a letter to, I wrote letters to some other people, but that didn't pan out. But I wrote a letter to Adrian Precision at the National Institutes of Health.
I see.
And for a long time, those were completely different times, of course, I have to say.
Yes.
86 as opposed to 2022 is a big difference. So anyhow, I wrote him a letter and then waited for the reply, and nothing was forthcoming. This was the height of the Cold War, of course. Also, the sending a letter from what was at that time Yugoslavia to the United States was, maybe there will be a reply, maybe not. So after a long wait, eventually I get a letter saying everything is ready. You can arrive on November the 1st, and this was like October.
I see.
So I was totally surprised at that time. I said, this is incredible. And of course there was no email, so we communicated via so-called telegrams. I'm not sure, do you recognize?
It's not a fax. Not even before fax.
I don't think there was a fax at that time.
Oh, really? Yeah. I see.
So this was a telegram that was sent by telegraph lines, and the communication had to be short because you paid by the sign.
I see.
And so we communicated. I bought a ticket, and he said, I will be waiting for you at the airport in Washington, D.C. And I will be wearing my Australian rancher hat. And I immediately went to the library and I'm looking for I have no idea what's Australian rancher hat. And eventually I found out something that looked like Australian rancher's hat. And that's how it started. That's how I left Slovenia or Yugoslavia at that time, and I went to the US. And I distinctly remember I was in the airplane. It was a intercontinental flight, of course. I think it was a DC-9 at that time. And as we were approaching, I didn't know that the airport in Washington, D.C. is called Dulles International Airport. So the flight attendant said, the PA message was, we are approaching the Dulles airport. And I was thinking, oh my God, this secretary, I told her Washington, not Dallas, Texas. And now I'm coming to Texas. And what do I do there? So anyhow, it turned out it's okay. And Washington has 2 airports, and unfortunately, Adrian Persijan went to the other airport to wait for me, so there was a little bit delay, this and that, but anyhow, this is how I arrived at the National Institutes of Health, which was the biggest scientific research institute in the US at that time. Biggest in the sense that they had the largest budget. Also for so-called intramural and extramural research. So they were also giving grants to other researchers that were not working at the NIH. And that budget was steadily increasing until the time of Clinton, and then eventually it started shrinking. But in that time it was absolutely, great to be there. They had a little museum of various discoveries that were made at that institute, including the discovery of the genetic code by Marshall Nirenberg. And I remember going to the museum and looking at all these famous people working at that time. So they were very strong in fundamental science, and there was a lot of fundamental research. They had a math department. In NIH. And some people, let's say, that worked at that time at NIH were Terrell Hill, who wrote the 2 statistical mechanics books. Introduction to StatMech and StatMech. So he was working there. And there was also Bob Zwanzig, of the Zwanzig non-equilibrium.
Was he there?
Yeah, he was there. Actually, I spoke with him a few times. Very nice man. Very nice man. So many other people which are completely fundamental. Also George Weiss, the editor of the Journal of Statistical Physics. He was there. Very, very theoretical person. Very kind and nice fellow. So I got a feeling, I am among my people, and I was able to proceed with theoretical physics. At the beginning, I was a little bit scared because the National Institutes of Health, I am a doctor, but not that kind of doctor, as they say. So anyhow, I was able to proceed with my studies, but not the line that I sort of had in mind for continuing my PhD work, but I started working on DNA.
You already studied?
Yes,
on
at
DNA
that
at the
time.
Yes.
So I never worked on specific biological macromolecules. This was the first opportunity to do that kind of work. And actually talking with Adrian, we decided maybe it's a good idea for me to start actually with experiments to get a real feeling for what is going on with DNA. So we did a lot, of course, with a lot of help, specifically from Don Rao, who was the prime experimentalist at that time in Building 10. I'm not sure whether you've ever been.
No, I haven't been.
So this is an enormous place. Enormous place. I think altogether it was more than 10,000, maybe 15,000 people were there. A lot of them were PhDs and so on. So we were in these huge buildings, which was supposed to be the biggest brick building in the world. I I'm not sure that that's true or not, but that's what I heard.
And
I was on the 9th floor. The lab was on the 9th floor. And let's say the lab of Dr. Fauci, who recently became a sort of a household name in connection with the COVID policies in the US, he was on 10th floor.
10th floor? I see.
And I met him many times in the elevator. I was, of course, a lowly postdoc at that time, and he was already a PI in the lab. But he always, he said hi to everybody in the elevator.
I see.
I remember him as a very nice and kind
very friendly person.
Very friendly person.
I see.
So I started working on DNA and this and that, DNA here, the DNA there. We published a few experimental papers, and then I continued working with on electrostatic problems related to DNA, on the structure of DNA mesophases. This is maybe the coolest work I did at that time. We discovered something that we refer to as the line hexatic phase. Of DNA. You know what a two-dimensional hexatic is, right? And DNA, if it's ordered in one direction, if you cut it perpendicular to that axis, you get a two-dimensional system, a quasi-two-dimensional system. And it behaves like a hexatic phase.
So did you do experiments?
Yeah, this was experimental. Yeah, very cool experiments. We collaborated with Alan Ruprecht from I think this is the Stockholm Technology Institute, the Karolinska Institute. And he was the world expert in preparing DNA samples. Of course, if you have the proper machinery, anybody can do the samples, the experiments, if somebody provides the samples. So that's the art part of the experiment. And he was able to procure this extremely oriented, almost ideally oriented samples of DNA that the whole world was using. All his samples for NMR, for X-rays, for neutron scattering, everything from his lab. And we were using those samples, and we saw some patterns in the X-ray diffraction that told us that possibly there is we are dealing with the hexatic phase. And of course the excitement was hexatics are two-dimensional phases, and here we have a three-dimensional system. How is this even possible? So we communicated with David Nelson at Harvard at that time. He is the discoverer, or he proposed the hexatic phase in two-dimensional system. So he visited us and he said, yeah, this totally looks like a hexatic phase, and there maybe is there are some theoretical hints from a few years ago that you could have a hexatic phase in well-ordered nematic polymers. Nematic polymers, like DNA. DNA makes a nematic, aligned polymers. And he said, why don't you call it a line hexatic phase? Because you have lines of DNA, oriented in one direction. And this is what we did. We called it a line hexatic phase, and this was a PNAS paper we published at that time, well received. The audience, of course, is not very big. I mean, how many people are interested in DNA mesophases? But it was a well-received work. I gave many talks on it, and later we tried to extend it and study it more in detail. And it turns out that the density of DNA at which you can see this kind of mesophases is relevant also for DNA packing in viruses, which is something that I'm doing for the past, 10 years.
Recently, right?
Yeah,
I see.
More recently.
I see.
So there's a connection between my recent interests and my interest from the times of NIH.
Was it connected?
Yeah,
I see.
Right.
I see. Right. Good.
Also, I, at that time I met many people that later on I sort of regularly communicated with from the field. But now everything's different, NIH has changed fundamentally, in the sense that it does not have that much fundamental science anymore.
Not anymore?
Yeah, I remember, when this started, everybody was supposed to give a short talk to the scientific director of that institute. So I should say National Institutes of Health. So it's many institutes. I think it's like 15 or something like that. And I was in one. Different institutes, but always one. So the scientific director of that particular institute wanted us to give a presentation of our work, and after you were done, so I don't know, 5 minutes, 10 minutes, quick overview, he would ask you one question: which disease are you going to cure?
I see.
And of course, many people didn't have an answer to that. We do fundamental science. As, and somebody asks you which disease are you going to cure. We are not that kind of doctors, right?
No.
So I didn't have an answer.
I see.
And many other people didn't have an answer. Many people left. There was a reshuffling of the labs, and some of them were closed down. So anyhow, at that time, I also got a position in Ljubljana.
I see.
Yeah, I became an assistant professor.
Oh, so how long were your postdoc period?
Postdoc was 3 years. Then I returned to Ljubljana. Those were very turbulent times. This is around 90s. At that time, Yugoslavia was falling apart. Slovenia proclaimed independence. So those were very, very turbulent times.
I see.
A little bit like what we see these days in Eastern Europe.
I see.
But I was also personally involved because I was from Slovenia / Yugoslavia, and there were all sorts of problems. I had a hiatus for 2 years, and then I continued as a postdoc in Sweden, in the chemical center of Lund, University of Lund. I was working with Bo Jensen, who is now a long time already retired.
I see.
And we did some simulations at that time. On also electrostatic systems. So the University of Lund, and specifically the Chemiecentrum, the Center for Chemistry, was well known at that time for the level of expertise they reached in terms of simulation of electrostatic systems. Electrostatic systems are of course complicated because Coulomb interaction is long range. So simulations are not trivial. You have to take that into account, and if you just make some cutoffs and so on, it never works.
I see.
So they developed a very sophisticated methodology to deal with long range interactions, and this is what I learned when working there. So after staying in Lund, I got an invitation again from NIH to return to NIH as a researcher, which I did. And I worked in the group of Adrian Precision as I guess my title at that time was like visiting researcher or adjunct researcher or something like that. I don't remember anymore. So at that time, we continued to work on DNA, and also those were the times when the so-called correlation effect in Coulomb systems became very popular, and many people were working on that. So, including myself, I started to work on that even before that time. The story is, if I remember correctly, that Dušan Bratko, who was another Slovenian from University of Ljubljana, he started simulating already in the 80s the inhomogeneous Coulomb fluids, so like electrolytes confined between two walls. Many people were simulating already bulk Coulomb fluids, bulk electrolytes, but this was confined electrolytes. And he found out that in some cases he got attractive interactions between two charged walls. And I think it was not clear at that time whether there's something wrong with the code or methodology is not appropriate. So is this a real result or is this an artifact? And later the Lund group started working a lot on the simulations of inhomogeneous Coulomb fluids, and there's a famous paper by Jönsson and Linse and Wernerström, if I'm not mistaken, and they definitively proved that you can have attractive interactions between equally charged bodies, of course, which is totally counterintuitive, and all the rage was how to explain this. And there were many people trying to get to solve this problem with various different methodologies, theoretical methodologies, and at the end I think the most successful approach was the one that was pioneered by actually Roland Netz, who was at that time in, I think, Munich. He sort of invented this strong coupling, weak coupling dichotomy, so weak coupling Poisson-Boltzmann, strong coupling expansion in the activity of the solution. And I was also working on that, so because in the context of DNA, the correlation interactions, Coulomb correlation interactions are extremely important.
I see.
And they lead to condensation of DNA. DNA is a stiff polymer, it's highly negatively charged, but it turns out that at some conditions, let's say if you add a polyvalent salt, they would condense.
I see.
So how do you explain that? And we were working a lot on that. I think Adrian Precision and myself were the first who started with the idea that this is not a Poisson-Boltzmann phenomenon, that this needs to be either fluctuations or correlations or something like that. And we wrote a paper on that, submitted it to Nature, and it was rejected. That this is not, you know, at that time people thought it's something else. So this was before Roland Netz came up with the strong coupling phenomenology. But later the development of strong coupling, it's not a fluctuational theory, it's a different fixed point in the parameter space, but nevertheless it's not Poisson-Boltzmann, so that much is clear. At that time there were some controversial papers, you know, people saying that, yeah, if you solve Poisson-Boltzmann equation in some weird geometry with some strange boundary conditions, you will get attractions, but that turned out to be completely false. So this was a huge event in DLVO interactions. In the theory of DLVO interactions, I think maybe comparable to the Deryaguin-Landau-Verwey-Overbeck first step to formulate interactions between charged soft matter. To realize that Poisson-Boltzmann is not definitive, that there are physically relevant conditions where you just cannot use this mean field theory.
I see.
And today I would say this is well acknowledged, but you know, today the simulations are not that expensive, anybody can do it, so if you can avoid any kind of analytical theory, that I guess today would be the preferred direction.
I see.
As opposed to some very, you know, heavy metal calculations. Very few people do that.
I see. So today this equal charge attraction is already established?
Absolutely, well established. So a while ago already the question is, when you go to real systems, not like you know, primitive model electrolytes, where you take into account water also and the structure of water, how does that relate to the correlation effect? That I would say is not entirely clear. And the coupling between the two, between electrostatic correlation interactions and water structure, maybe is something that is still a developing field. So I wouldn't say that this is, you know, that people have solved this problem and everything is understood.
I see.
And specifically, of course, that's important for membrane-membrane interactions and for DNA-DNA interactions.
Okay, and so after you returned to NIH, what happened?
After I returned to NIH, I stayed there for a few years. And I think 3, maybe 4 years. And then I got an invitation from the University of Ljubljana if I would be interested to become a professor there. And I thought a bit, and I decided that maybe that would be a good idea for me. But I was already very much involved with the research at NIH, so I didn't want to entirely break with them and fully move to the University of Ljubljana. So I retained some kind of engagement in the US, and I was returning during each academic holidays at the University of Ljubljana. I returned to the US and worked with the same people.
That's why you had very strong connection
correct,
with NIH.
Correct. We did a lot of work together, all sorts of work related to the interactions between macromolecules, soft matter, and at the end also the physics of viruses. This was the last thing that I started doing while at NIH. I did, when I was in Lund, I started doing simulations, but then later I did not myself pursue this direction. I did not see myself as a simulator, but I always collaborated with people doing simulations. But myself, after that work on polyelectrolytes, so I was simulating polyelectrolytes and so-called electrostatic bridging interactions between nanoparticles and so on. I never did simulations of my own.
I see. And so after you returned to University of Ljubljana, so you were there for quite
a long time, of course, until
you
I retired.
Meanwhile,
I also had a visiting position at the University of Massachusetts in Amherst. This is connected with the fact that my good friend and collaborator Adrian Precision moved there, and I decided that I will spend time with him and continue our collaboration. So this is what was going on until he eventually retired there, and then I didn't continue collaborating with that university. I also had a collaboration with the Case Western University in Cleveland. I collaborated at that time with Roger French. Roger he has a different background. He is an experimenter in condensed matter physics. He worked on, let's say, ceramics and various application of various spectroscopic techniques in ceramics. And we had a DOE grant at that time involving also Wei-Yim Ching, who was doing theory of condensed matter. And later some of my work was connected also either with Roger or with Wei-Yim. And with Wei-Yim I continue collaborating until these days. What we are doing these days is we are applying heavy ab initio calculations to the S protein and other viral proteins in the COVID era. We were looking at the various mutations, and as you probably know, each successful mutation had a higher number of positively charged amino acids. It's not exactly clear why this is the case, but these are some of the problems that we worked on. I had also various PhD students and postdocs with whom I worked on different aspects of soft matter, including polymer theory, polyelectrolyte theory, nanoparticles, stability of nanoparticles, membrane biophysics, fundamental interactions, correlation effects, structural effects, and so on.
Okay, so I'm also curious about your country, Slovenia, and can you briefly explain about Institute Joseph Stefan and also University of Ljubljana? Unfortunately, I haven't been to your country.
You haven't been, it's a pity.
So I'm very curious.
There are a few Japanese scientists that are, I can't recall the names right now, but mostly in the field of liquid crystal physics. Liquid crystal groups is very active and well recognized. So the Josef Stefan Institute, it's of course related to Josef Stefan, the Stefan Boltzmann law, and so on. He started as a Slovenian poet He was a poet.
Poet?
Yeah, he wrote poetry in Slovenian language. He was not particularly successful. As far as I can say, it's good poetry, but the people at that time thought that he sucks. So there's a very well-known literary critic at that time who wrote a scathing review of his poetry, and Stefan decided, this is it, I'm out of here. And he moved to Vienna, where he became, among other things, also the teacher of Boltzmann. So a very important person, an important Slovenian scientist. And the Josef Stefan Institute, if I remember correctly, was so it was established after the war, maybe in the 50s.
I see.
And at that time, interestingly enough, the director of that institute was Anton Peterlin. I'm not sure whether you recognize this name, but Anton Peterlin was a very famous polymer scientist. Yeah, this is before the Gen, so mostly people don't recognize this name anymore because the people before the Gen, polymer science is before the Gen and after the Gen, and scaling concepts and so on. So this was the pre-Gen polymer physics. Yeah, and he was working on all sorts of cool problems, an extremely important person. And I didn't know about that before because later he emigrated from Slovenia. There were also some kind of political problems, if I understand correctly, connected with the fact that Yugoslavia at that time was trying to establish a nuclear program, and he wasn't particularly happy with that. So he moved out first to Germany and then to the US. And he was high-level scientist at the National Bureau of Standards, which today is called National Institute of Standard and Technology, NIST. Yeah? And when I was a postdoc in Washington, D.C., somebody invited me to give a talk at NIST, and I went there, and I was waiting for him in the lobby of NIST, and I was walking around, and then I saw a huge portrait in oil. And I look at the name and it says Anton Peterlin, Professor Emeritus, or Scientist Emeritus, something like that. And I said, wow, I know nothing about this guy. How does this even happen? And then I explored a bit the situation. I realized he's was the first director of the Josef Stefan. Yeah, and he had, his vision was this should be a polymer institute.
Oh, really?
Yeah, he was trying to pursue this direction, but he was apparently unsuccessful. There were other forces at that time making it not a polymer institute, but a nuclear physics institute.
I see.
Polymer science was not, chic at that time.
I see. Was he a theoretician?
He was a theoretician.
I see.
Yeah.
I see.
So he was actually. This also I didn't know. He's the person that first interpreted and came up with the number for DNA persistence line. This was in the 50s, and everybody was scattering something on DNA. So Rosalind Franklin was scattering X-rays, and Peterlin and his group were scattering light on DNA. Yeah? So of course there's a difference in scale. With X-rays you can see local arrangement of the atoms, and with light scattering you can see fluctuations in the chain. And he tried to interpret the experiments. Experiments are not done by him, but he tried to interpret them and came up with a number for the DNA persistence length, which was like around I think 500 angstroms, 50 nanometers, which is very close to what we consider today to be the canonical persistence length of DNA. And he had a Nature paper in 53, which is the same year as DNA. Watson and Crick. A few months difference. That just tells you, everybody really was scattering something on DNA.
So he didn't know the precise structure, but knew the.
I think not, but he knew that it's a polymer, a polyelectrolyte, and he knew that you can scatter light on it, and to interpret the scattering of light, you have to model the chain as a worm-like chain, which is what he did in some approximation, and was able to fit the data. Quite successfully.
Oh, that's interesting.
Yeah. So I found that out when I was at NIH and I was listening to what's his name, maybe Hagerman, who worked on DNA also and wrote a very long review on elastic properties of DNA. And Peterlini is like the first reference. I knew nothing about that. You know, it's 53, so yeah, you have to be motivated to go and look what's in that paper. But I did. So this is my connection also with the first director of the Institute, Josef Stefan, where I worked at the theoretical physics department. So I was, I think I was the first one, or maybe, possibly the first one to start a group. Actually, no. Sasha Sutina and Boštjan Žeč, my PhD mentors, were the first to start a biophysics group, which I continued then for as long as I stayed at Josef Stefan Institute. And also I got actually it was the reverse. First I got a position at the University of Ljubljana, what was called the Faculty of Mathematics and Physics. I became assistant professor and then associate and full professor, and also a position at the Josef Stefan Institute as a researcher.
So you have the position in
this
both
is
institutions?
Actually this is quite usual. We have like double position, not a full position. So we have a full position at the university and a small position at the Stefan Institute, because Stefan Institute had better, at least at that time, had better capabilities for students and also experimental work and so on.
As far as I know, these two institutions are located very nearby.
Across the road.
Just across the road. I see. So it's very close. And
very close.
People also interact between these two.
Some people. So it's very interesting. You know, physicists do interact with one another, but other scientists, where the main departments are not so close to the institute, they usually did not interact, or maybe not to the same extent.
I see. Okay, so then let me ask you the next question. So, why did you come into the field of soft matter physics, and who were the influential scientists for you? You already mentioned some of them, but maybe,
so as I told you, my original field was macromolecular interactions, but not specifically between soft materials, but in general. So the first time I started working on soft matter was when I moved to NIH as a postdoc, and then I somehow continued all the time, but different aspects of soft matter. Then I didn't mention yet my work together with David Endelman in Israel, whom of course you also know very well. We established our own direction specifically in the theory of electrostatic interactions, various aspects of these interactions with him and with his various students in Israel. I also visited Israel, specifically the Tel Aviv University, many times, gave lectures there. I was also an invited fellow, and so this is a very deep collaboration, and a lot of aspects of soft matter, of course, were studied when I was in Israel and when I collaborated with David. So who probably I would need to say that maybe Adrian Precision is the person who sort of introduced me to soft matter, or bio-soft matter I should say, not really soft matter but biological soft matter. And also the fact that in the lab people like Don Rao were doing sophisticated experiments with various polymers. This also sort of set me on the way to study different aspects of soft matter. I think it was a good decision. Before, maybe when I was an undergrad, I was thinking a little bit, maybe I should do general relativity or something like that, maybe fundamental interactions in like field theory of fundamental interactions. But I lost interest somehow. It was, I don't know, I never got really hot about subjects that I thought everybody is working on. This didn't, didn't attract me.
I see.
It had to be something that was not that popular. And at that time, maybe soft matter was not so popular.
Maybe not.
Few people who worked on it, but the situation improved almost every year. There was more and more sophisticated work done on soft matter. People realized, these are incredible systems that you can apply very sophisticated theoretical methods to.
Yes.
And specifically also de Gennes' contribution to polymer physics, polyelectrolyte physics, and many other people. Edwards also.
Yes.
They made substantial contribution to what would be considered today as soft matter physics.
Yes. Okay, and you already mentioned, but you have been actively working on the physics of DNA and viruses.
Yes.
So why do they attract you
Well,
so
DNA,
much?
of course,
Yes.
DNA is DNA. And today, you cannot ask why do you work on viruses, because everybody does.
Yes. Right now, yes.
Yeah, right now.
Yes.
So, a little story from my time in China a few years ago. I submitted a proposal at NSF, like you are doing now.
Yes.
And the title was Physical Virology, and this was before the pandemic. And I made a presentation, and people were not particularly interested. Why viruses? Why is this physics? How can physics say anything about viruses? So I said, well, okay, not interesting. But then next year I made the same presentation after the pandemic started, and they were very interested what this guy has to say. So of course now.
But when you started, the situation was different, right?
No, still, I started because I was genuinely interested in viruses. I was following the work of Bill Gelbart at UCLA. He did some spectacular work on not only physics of viruses, but actually sophisticated experiments with viruses. And there are beautiful physics problems, let's say the symmetry of the viral shell and so on, where physicists were involved from the start. Right. So Crick and Watson, first hypothesis that you have icosahedral symmetry in viral shells, and then various models of packing of proteins in viral shells, which are related and highly dependent on physical models and so on. So viruses are an interesting, we would call today nanomaterials, because quintessentially they are on the nanoscale, a few tens of nanometers.
I see.
So they are nanomaterials, and they embody very interesting aspects of physics.
I see.
Okay. Don Casper, let's say, who was working on the symmetry of viral capsids and the environment of proteins inside such a in the shell of the capsid. He was also a physicist, and those were also a little anecdote, maybe connected again with China. When I was in what is the province with Guizhou? Is that the province? Guiyang. Guizhou. Okay. There was a trip to Guizhou, and we were, visiting various places, and we also stopped at the museum. So we were looking at this museum, and all of a sudden, in this museum, I saw a whole wall dedicated to a certain person by the name, I think, of Pollard, who was a missionary in Guizhou, in some village in Guizhou. And it said in the description that this is an American missionary, who during some kind of an epidemic got a vaccine from the US and vaccinated the local population and did some, very important humanitarian work and so on. And his son later continued his education in the US. And I was thinking, Pollard, where have I seen this name? And then I remembered that at my home, at my house, I have a book with the title Physical Virology, written by somebody with the last name Pollard. So I started digging into this, and I realized this Pollard, who wrote the Physical Virology book, is the son of this missionary in Guizhou that I saw in the museum of Guiyang.
I see.
So, interestingly enough, at that time, I was already working in physical virology. This is an older book. It's not recent, but it's the first book that talks about physical virology, which later, when do you know a subject in physics is like mature? When you have a Gordon conference with that name, of course. And that was some years before. I came to China. The Gordon conferences in physical virology started in the US, and they continue. Yeah. So this guy Pollard, the son of a missionary in Guizhou, China, was the person who wrote the first book on physical virology.
I see. That's very interesting.
Yeah.
That's very interesting. I see.
You cannot buy it anymore. I mean, you can buy it in second-hand bookstores.
Okay. Do you have it?
I have it.
Good. And when was that book exactly?
So a good question. Yeah. So when was that? Probably maybe 80s.
80s? Yeah. I see. Okay. This is the time when you also started to
work. But at that time nobody was talking about the physical virology.
I see. Good. Okay, so then maybe next question. Okay, so you also worked a lot on fluctuation-induced interactions, right? And can you explain me why they are interesting for you?
For the interesting part is that fluctuations are a little bit like you don't need to have a source. Like if you have charges. The source are the charges, and the interaction is a consequence of that source. And if you have fluctuations, you don't need to have any net charges. You can have neutral bodies, and yet there are charge fluctuations which lead to interactions. And my first encounter with fluctuations was when I was during my PhD studies. I was studying the Lifshitz theory of van der Waals interactions, and that is, of course, a fully fluctuation theory. It's there's no mean field Lifshitz theory. It's just fluctuations. So I got a taste for fluctuations at that time, and I was trying to find out what would be some other cases where the fluctuations were important. And for me, the really important step was to realize that you can get the Lifshitz theory, or at least the so-called zero Matsubara frequency term in the Lifshitz theory, if you look at the fluctuations around the Poisson-Boltzmann equation. That gives you terms which are exactly the same as the zero frequency Lifshitz term. So that's the connection. And the other connection was a little bit later when I worked with David Endelman, as I told you, and we worked on the surface tension of electrolytes. And we realized that what is referred to as the Onsager-Samaras surface tension of electrolytes, and for which people, but not only people, Onsager thought that it's a mean field result. It actually is a fluctuation result.
Is it?
And you can see that quite clearly if you start with the partition function and go through all the steps. You realize that the calculation that you do is not a mean field calculation. Actually, mean field gives you zero.
Okay.
So it has to be fluctuation.
I see.
And various other types of fluctuations, also membrane fluctuations and the DNA, conformational fluctuations were on my map of interest, and I tried to pursue some of these interests throughout my career.
I see. Okay. Then let me ask slightly different question. So you moved to China in 2017?
Not exactly. I think 2018.
18. Okay. Yeah. And it must have been a big change for you.
Oh, definitely. I had no intention
why did you
ever to move to China. China was not.
China?
Yeah, China was not on my map for some reason.
For some reason.
Yeah, many places were not on my map. But I really wasn't ever thinking that I could work in China. Possibly also because my conceptions were, mostly a consequence of reading Western media, and the image of China that you get from that is just weird. So it was not on my map. But then a student of mine, my first PhD student, got a position in China, and he was, Yure.
Yure, I see.
Yure Dobnika. He got a position in China. It's a longer story. I don't want to go into details. And he was inviting me all the time. Why don't you visit? Why don't you visit? And eventually I said, why not?
Why not?
Let's go and see. I didn't have any expectations. I didn't, I didn't think that would lead to anything. But I visited China and I was totally impressed. First, I was impressed by the level of science, by the way science is treated, all the different universities, the different institutes and so on. And I realized, man, this is not bad at all. I could work here. And then somebody asked me, would you be interested to maybe, part-time or whatever, to spend some time here? And then I started thinking, and at the same time I was also approaching, years are passing by, and eventually you realize you're old. Happens to everybody.
Yes.
So I realized, okay, I'm approaching my retirement, and maybe it's a good time for me to think it over, see what would I be doing for the next N+1 years. And I thought maybe this would be an interesting step for me, not only continuing my scientific career, but learning about the world, in general, learning more. I'm interested not only in science, I'm interested in all sorts of things.
Cultural things
all sorts of things, and I just want to get a good impression of how this planet looks like, what's going on. So to me, this international collaboration aspect of science was always very, very important.
I see.
I saw it as a very fundamental part of science, that we can fly for 12 hours, and then you go out and talk with people like there's no need to explain what Euler-Lagrange equations are, what fluctuations are. Everybody's on the same page with you, and that's fantastic with science. I don't think there are other endeavors of humanity where you can switch between different, ethnic categories or countries or continents in the same way that you can do with science. I think you agree, right?
Yes.
You also came to China. It's not like here science is some different science that you need to learn. It's the same science, the same jargon, the same equation. Everything is pretty much the same. So eventually, I ended up spending some time here. It's of course a more convoluted story, this and that, but anyhow, that's what it is, and I'm still here.
And you have been in China already like 3 or 4 years?
Yes, of course you have to take out almost a year that I spent in Thailand because of the epidemic and so on, but that's a separate story.
And how is your life in China in general?
Fine, I like, absolutely.
And you're learning also?
I learn Chinese. This is like a challenge for me, at my advanced age, to start learning a new language. But I'm perfectly up to it, and I find it very interesting to compare the Indo-European structure of language with the Asian structure of language, and it enormously amplified my understanding of what the humans are all about. How come you can say something in this way when you come from that background, but in this background you say it completely differently, and yet the meaning somehow is the same. And you can recognize the meaning. So that also I find fascinating, and I enjoy very much learning Chinese, and I have a good teacher and so on.
I'm very impressed that you are also learning how to write Chinese characters.
This is, I have to tell you that when I arrived in China, I couldn't remember any character And it was really weird. I thought, maybe this is dementia already or whatever. The guy's old, he can't remember anything. So I looked at a Chinese character, and if I look at Latin characters or even very complicated drawings, I close my eyes and in my mind's eye I can see it. I can reproduce in my mind what I was looking at with my eyes. But with Chinese character there was nothing. I closed my eyes, zero. Zero. Nothing.
I see.
And I thought, I cannot learn this language. This is impossible. And also the names were very difficult. But then when my friends visited, I realized they had exactly the same problem.
I see.
Everybody had the same problems. You cannot remember the names and you cannot remember the characters. And I think you really have to build some kind of additional connections in the brain, some kind of new synapses to be able to recognize the characters. Now, of course, I close my eyes and I realize the parts that compose the character, and that, of course, I can remember. And I close my eyes, I can see it. And then I can write it. But before, this was for a long time, more than maybe a year, I was not able to reproduce a character. I had to go line by line. When I learned the characters, I had to line by line. I was writing it. There was no other way.
But that's how also Japanese people learn Chinese characters when we are a small child. Yes.
But the way you do that and the way you express yourself in Indo-European languages, I think it's fascinating to compare with the Chinese language.
Yes.
I don't know other Asian languages. This is the only one I know.
Yes.
So also, linguistically, I'm very interested in languages, and I don't know Thai, but I spent this year in Thailand during the epidemic, and I realized how connected the Asian cultures are. Let's say in Chinese, I think the expression you use for so if you are how do you say careful.
Careful?
You say 小心, which means little heart. And in Thai language, it's how is it? Jai Noi, which is completely different. Of course, no relation with Chai Xin. But it means exactly the same thing. It means little heart.
Little heart.
And this I found, wow, how can this be? That two completely unrelated phrases actually deep down are the same thing. And this, I would say, is the general sort of Asian culture, like European cultures. We share a lot of the expressions that are related, even if they don't sound the same. They might be a different word, but the origin or the actual meaning is the same. So this, I think, is the same in Asia, in Asian languages. But of course, I'm not, I cannot say deeply acquainted with Asian languages.
So of course in Japan we all imported many Chinese culture in the past. So there are many things in common. But still, I have to make effort to learn Chinese.
So I also have a connection with Japan. My very good friend, old friend, she was learning Japanese. There is a Japanese language department in Ljubljana.
Oh, is there? I see.
Right, and they had courses of Japanese, and many people were attending that.
I see.
I also remember the first time I tasted Japanese food. Probably, I was a kid at that time.
Oh.
Yeah, and my parents took me to a restaurant, and they had a week of Japanese food.
Oh, really? Did you enjoy?
And not really.
Not really.
But the most important difference was that, at the table next to ours, there were other people sitting and eating, I don't know, sushi, sashimi, whatever, miso soup, and so on. And when they were finished, the father called the waiter and sort of sotto voce, not very loudly, asked him, okay, but do you also have some Wiener Schnitzel? Japanese food, fine, but now some real food.
Okay. Okay, so now you are visiting Wenzhou Institute,
yes.
And can you explain your relation to this institute, because your official position
officially I have a position
in
UCAS in Beijing, and
what's your role here in Wenzhou?
Yeah, so I'm friends with the director of the Wenzhou Institute, or the scientific director, Ye Fengfu, and we had many and still have many common interests. And he said, "Why don't you visit Wenzhou?" And I know from before that in China it's a little bit tricky. What is the level of the institute? I visited some institute. I kind of saw that this doesn't have a future. It was not. I don't know. Really. People were not fully dedicated, or that you just realize this will not continue. But some of the institutes, especially newly developed institutes, are absolutely very strong in terms of researchers that they employ there. Also, they have a vision and so on. So I wondered, okay, we will see what's going on in Wenzhou. And I was totally floored when I saw the institute, a very serious institute. They obviously have a good support of the local government.
Yes.
There's good people at the institute, including you, of course.
Thank you very much. Yes.
Your colleagues, and Professor Masa Doi, who is as we both know, of course, one of the fathers of polymer physics, now has a position here at the institute. So I realized this is serious. It would be nice if I could visit. And Professor Ye was kind enough to make it possible for me. And now I spend like a few months per year in Wenzhou.
Yes.
The weather could be better now.
The weather, yes. So yes, please come to Wenzhou as much as possible in the future. Okay. And I'm coming to the coming close to the end. And so this is just a general question. But what kind of scientific problem do you think will be important in the future? Maybe in soft matter or biosoft matter, or what do you?
Yeah, I certainly think that biomatter will not lose its importance. It already is important. And I'm sure it will not lose its importance. And maybe connection with sophisticated technologies is something that we should all keep in our minds, that this is worthwhile studying and investing some time into this research. Also, maybe it's easier to get funding if you work on bio-related matters. At least it used to be. Now, it's of course funding is problematic anywhere for anything you do. But I think it's still better than maybe some other research areas. And it's just interesting because being a physicist, you don't have this biological way of thinking, and I think that allows you to make a contribution which is specifically stems from the physical point of view, maybe even physical formalism and so on, that otherwise would not be present. Biologists think differently.
Yes.
This has always been my, I cannot say problem, but I realize that they think differently. They think very much in terms of images.
I see.
So you will never impress a biologist by talking about free energy. Yes. But if you show some movie, even if it has nothing to do with actual behavior, but you show some molecules moving around, they will be very prone to see that as a major advance in the field. So I would suggest. So this is what I think. Bio-related soft matter definitely has a future.
I see.
And also young people should be. Should be admonished to go in that direction.
I see. Okay, good. And maybe this is the final question. So I think most of the audience of this YouTube channel is Japanese actually.
Yeah.
And do you have any messages for young Japanese scientists or researchers?
Yeah, I don't know that many, so it's difficult to say.
Yeah.
But of course, probably they are in the same position as other young researchers elsewhere in the world. Of course, maybe the message is not altogether good. It's difficult to find jobs in science, but then it's difficult to find jobs anywhere in these times. So maybe some kind of a technological orientation based on soft matter physics and biomaterials is something that would give you an advantage in edge when you look for jobs.
I see.
Be them academic jobs or more applied jobs in some sophisticated industry.
Maybe not only in Japan, but in
everywhere.
Everywhere.
Yeah.
I see.
Japan situation in Japan, of course, I don't know that well or don't know at all. I know some very good Japanese literature, but apart from that,
I see.
Yeah,
Okay.
not a whole lot.
Okay, so Rudy, so thank you very much for your kind answers, and I'm very happy that you came to my channel and
thank you for inviting me
and answered many questions. Thank you very much.